This is a production of Cornell University.
Okay, thank you. So we all know, we all try to eat to maintain a healthy and diverse diet, but this is not always easy. Although the nutrient deficiencies are less common in developed nations, surprisingly high proportions of the US population still do not obtain the daily amount recommended for several nutrients, including vitamin E. So what if I tell you that we could help with that by increasing the nutritional quality of sweet corn, which I bet many of you like? So my project is trying to understand the mechanism of vitamin accumulation in order to convert the locally adapted sweet corn lines into dark yellow grain with high provitamin A and vitamin E. So carotenoids are compounds produced by plants, and some of them are provitamin A, while tocochromonols are vitamin E. So there are several compounds within each class. So from now on, I'm going to be using the colors orange and green to distinguish between them. As antioxidants, they might decrease the risk of several health complications, including cardiovascular disease, age-related macular degeneration, which can cause blindness, and specific cancers. A study showed that about 26 to 41% of US population has low levels of vitamin E, and this may be associated with increased risk of cardiovascular disease. Also, age-related macular degeneration affects about 25% of people of 65 years old and older. And Northeast is the region with the highest proportion of people over that age. So this demonstrates the importance of studies to increase the vitamin in the food crops. So sweet corn, because it's widely consumed, this could be a good candidate for biofortification, which is the increasing nutritional quality of food crops by breeding. Sweet corn is the third mostly consumed vegetable in the US. It is estimated that each person eats about 11 kilograms of sweet corn a year. And New York is one of the most important states. It is among the top 5 states in production and value of fresh sweet corn. And also it is the second state with the highest number of fresh markets. And this is a great opportunity for address common dietary deficiencies. Biosynthetic pathways for these compounds are well characterized. From here, the main ideas to remember are that alpha and beta carotene, as well as beta cryptoxanthin, are provitamin A. Leucine and zeaxanthin are associated with reduced risk of age-related macular degeneration, and alpha tocopherol has the highest vitamin E activity. Also, the enzymes denoted by the stars were previously reported to be associated with increase of some of these compounds. And this information could be used to improve sweet corn as well. However, when you look at the genetic distance of sweet corn to the field corn, you can see that sweet corn is a distant breeding pool, and it has also less variability. Also, the nutrient analysis of sweet corn needs to be done during the fresh stage, which is when sweet corn is eaten as a vegetable. So our main question here is, are the genes responsible for increasing vitamins the same at both stages? And are the favorable alleles found in field corn also present in our panel? So the goal for my project is to understand the mechanism of the nutritional quality nutritional traits in fresh sweet corn kernels. In order to achieve that, we're trying to identify causative genes and favorable alleles and develop and validate marker-based prediction models. In order to assess the natural variation for these compounds, we used a panel with 400 inbred lines from public breeding programs across the U.S. We planted 2 years, and then we harvest at eating stage, which is about 21 days after pollination. So the ears were then fresh frozen, shelled, and they were later ground and kept in a -80 freezer. And the carotenoids and tocochromanals were measured using HPLC. Using these phenotypes together with genotypes from GBS, we performed a genome-wide association study. And then for alpha tocopherol, which is the compound with greatest vitamin E activity, we found a significant association with markers in chromosome 5, which is close to this gene, VTE4, which is located, it encodes an enzyme located at the end of the pathway, right before the compound. And this candidate gene was previously reported in field corn as well. Also for delta to gamma tocotrienol, we also got a significant hits in chromosome 5. However, these markers are close to VTE1, which, as you can see here, in encodes enzymes, they are in both branches that goes to the compounds. When looking at genetic correlation between the compounds, we can see that some of them are highly correlated, which is shown by the darker orange in this case. We could use this extra information to provided by the cross-trait correlation in a multivariate GWAS in order to increase the power to identify associations. And also a joint analysis would be more consistent with the biology when a single variant is associated with multiple traits. So we did the same analysis but using multivariate GWAS, and instead of using the ratio of the two compounds, we are using both together. And then after controlling for kernel type, we still get the VTE1, the same hit that I mentioned before, but we also have a new association with this gene that is encodes an enzyme like from the precursors. And this was not previously reported for tocochromanals. And this also makes sense because since if you think like by having a favorite allele here, you can increase the substrate for the downstream compounds. So in summary, we identified genes, candidate genes, 3 candidate genes as I mentioned, and being this one a new association. These are all for tocochromanals. And we also found one association for carotenoids. But I just would like to mention that carotenoids, we only have 1 year of data so far, while tocochromanals, we have the 2 years already. And then we now expect to identify the best variants within those regions and use that to select the best performing lines to be used as a parents. We also use genomic prediction using genome-wide markers to calculate breeding values for each of those traits. We used a ridge regression BLUP, and then by doing 5-fold cross-validation, we got accuracies as high as 0.61 for this compound, or provitamin A had accuracy of 0.4 and vitamin E 0.35, for example. So the lines with the highest breeding values for complementary traits, such as, so we chose traits with high vitamin E and high provitamin A, for example, and then we crossed them last year. And the seeds generated from that, we are going to be planting this coming season to start validating the models and to start the breeding population. So in summary, we found different hits for some of the compounds, being this one a new association, we already started making the crosses last year, and then this is the first step to start increasing the vitamins in sweet corn, going from white with low vitamin A, for example, and to dark yellow lines with more provitamin A, and hopefully vitamin E as well. I would like to thank everybody that helped in my project, especially the Gore lab. Thank you for your attention, and I will be happy to answer any questions you may have.
So there are different colors of sweet corn available already. Do you have a sense for how popular darker yellow kernels are over white kernels? I don't have a sense of what goal temperature they use here in the US.
So I don't have a right answer for that because it's pretty subjective. But what I understand is that if you start telling people that that's gonna make them better, like it's better because it has more vitamins, and in theory it shouldn't change anything else, like the taste. We are trying to still make good lines, so I think they would be willing to change. And even though if they prefer like white sweet corn, I believe they would change if it's better for their health.
How much of the variance is controlled by those VTE1 and VTE4? I mean, are those like huge things that are turning you directly from like yellow, or is that pretty small relative to the other smaller things?
So there was, in the past for field corn, it was found was a really high effect. And then we didn't find as high as so I don't think we have as many variation as the field corn, but it's still we have really big hits for those genes, so they are still controlling most of the variation.
Just to clarify here, the criminals are colorless.
Yeah, I was wondering about how much of your variation in your vitamin E or A is controlled by those.
Oh, I thought you said color has variance under common.
Probably did say that.
Yeah. So those are colorless.
Okay, no more questions. Our next presenter will be Rachel.
This has been a production of Cornell University, on the web at cornell.edu.